Good afternoon, and thank you all for coming. My name is Paula, and I'm a PhD student from Brazil. Today, I'll be presenting to you part of my PhD in collaboration with the postdoc Ayana Martins and my supervisor Marcus Aguirre. Plant-animal interactions can show substantial variation in the outcome of their interaction, and so this variation in the outcome of the interaction can range from negative to positive. So if we think about a frugivore that consumes fruits, this bird can consume poop without dispersing seeds. Even among birds that do disperse seeds, there can be variation in the dispersal distance, which can affect the fitness and seedlings and probability of seedling. If we think about pollinators, there's also variation in the efficiency of pollination between different pollinators. And in extreme cases, pollinators can actually act as nectar robbers. There's also a specific type of pollination, which is a nursery pollination, in which insects also lay eggs in the flowers. And that can, and so the larvae that develops within those flowers would eat part of the seeds. For all of these types of interactions, there are specific costs and benefits, and the balance between those two will determine if this interaction will be mutualistic or antagonistic. Even between the same pair of interacting species, there can be variation in the outcome of the interaction between years and between communities. And if we think about the species being distributed in different communities, gene flow between those communities would influence their coevolution, and hence their patterns of evolution and coevolution. And so I am specifically interested in species in which there's variation in the outcome of the interaction and how gene flow between communities can influence their coevolution. So with that in mind, the question I'm interested in answering is how meta-community structure and gene flow can affect coevolution between a pair of interacting species. In this scenario, there are communities in which the interaction will be antagonistic and other communities in which the interactions will mainly be mutualistic. And so coevolution between species will lead to changes in the traits of interacting species. Those changes in traits can lead to different degrees of matching and mismatching between species. Here, these are two pictures of gray moths and their Lithophragma host plants. And so some of the traits involved in this interaction include the proboscides of the moth and the depth of the flower. And these two traits are involved in nectaring and pollination. There's also another trait involved in this interaction, which is abdomen length and ovary depth. And these two traits are involved in oviposition. This is an example of a nursery pollinator. So all these traits, in a simplistic way, we can describe them as two alleles. So, for a pollinator species, we can think that a pollinator has alleles capital A and small a, and a plant has alleles capital B and small b. And the interaction happens when those two alleles match. So individuals that have allele capital A will interact with plant individuals that have allele capital B, and the same thing for the other allele. And we can calculate the mismatch between those two species as the absolute difference between those two allele frequencies. So for the pollinator species, the frequency of allele capital A can be described as X, and for the plant species, the frequency of allele B can be described as Y. And considering that the interaction happens when these alleles match, we can follow how these alleles changes over time, given the fitness that this interaction will provide to those individuals. So in this graph, on the x-axis, we have the generations, and on the y-axis, we have allele frequencies over time. The orange curve describes changes in allele frequencies for the pollinator species, and for this scenario, what happens is allele A is fixed. And in the green line, we have the plant species, and in this scenario, the plants remain polymorphic. And the estimation of mismatch will be the absolute difference between those two curves. And so if we think about mutualistic and antagonistic interactions, here on this blue square, I'm showing what happened within this blue square. I'm representing what's the expectation when a community in a community in species interact in a mutualistic way. So what happens is that over time there will be a fixation of alleles for both species. In an antagonistic community, alleles will oscillate, which I'm representing here in this red circle. Alleles will oscillate over time, and that will lead to polymorphic populations. So the expectation for isolated communities is that mutualisms will favor fixation of alleles, and antagonisms will favor polymorphism. But however, communities can be connected, and the interplay between gene flow and interaction strength will change genotypical patterns found within communities. And in these scenarios, for mutualistic communities, there can be polymorphic species, and in antagonistic communities, we can find fixation of alleles. So all this work was done considering two different communities connected by gene flow. And my goal here was to understand how meta-community structure could affect coevolution, and also to propose a new way of, an additional way of estimating coevolution, taking into account the role of interspecific polymorphism and interspecific interaction between species. So interspecific polymorphism can be estimated as the variance between between alleles within a species. So here we would have so to calculate that, we simply multiply allele frequencies within a species and sum it between the two species to have an estimation for a community of the degree of polymorphism. This metric can range from 0, when there's no polymorphisms and alleles are fixed for both species, and has a maximum value of 0.5, when in both species there's polymorphism, and so the frequency of each allele is 0.5. And so an interspecific interaction is what will define the species fitnesses, and a way to estimate that is to multiply the frequencies of these matching alleles between the two species. And this gives an estimation of the suitability of finding a partner to interact with. So in this matching allele model, X would interact with Y, and individuals that have 1 minus X would interact with individuals that have allele 1 minus Y. This metric ranges from 0, when non-matching alleles are fixed for different species, and has a maximum value of 1, when the same allele matches for both species, and so interaction would be maximum. And it has an intermediate value of 0.5, either when both species have high polymorphism, or when one of the species has a fixed allele and the other one has a maximum polymorphism, which means that half of the time individuals would find a partner that would have a fitting match to themselves. And so coevolution, we can investigate coevolution by the combined effect of the interspecific polymorphism and the interspecific allele matching. And so we can plot this, we can create a parametric plot considering these two metrics. Here in the y-axis, we have the polymorphisms ranging from 0 to 0.5. On the y-axis, we have the interspecific interaction. And we can map the expectations from mutualistic and antagonistic communities within this parametric plot. So for mutualistic communities in which the expectation is fixation of alleles, what happens is that interaction will be maximum and polymorphisms will be minimum. And that can be seen, that can be mapped as the blue region up in the corner of the parametric plot. For antagonistic communities, which has the expectation of oscillation and polymorphism between alleles, that will lead to a maximum polymorphisms and an intermediate value of interspecific interaction. And this can be mapped in that red region here in the map. But gene flow between communities can change those genotypic patterns. And so we investigated how the degree of gene flow could change those expectations. So here, this color bar indicates the degree of gene flows that we investigated. And each of these dots indicate the values for isolated community when there's no gene flow. And again, the blue square is the mutualistic community, and the red square is the antagonistic community. So as gene flow increases, each of these dots represent a different value of gene flow and how that affects coevolution between those two communities. So the genotypic patterns change as gene flow increases, decreasing interaction in both communities and increasing polymorphisms in the mutualistic communities, and then finally, as gene flow increases, both communities will reach a polymorphic state. So in this scenario, with two communities, high levels of gene flow will lead populations to be polymorphic. But frequently, populations are distributed more than two communities. So when we consider three communities, with a central community and two with a central antagonistic community and two peripheral communities, what happens when gene flow increases? So in the middle, we have the antagonistic community. It remains polymorphic, regardless of the degree of gene flow. For the peripheral mutualistic communities, what happens is, as gene flow increases, it rapidly reaches a polymorphic state. So with this spatial structure, for low levels of gene flow, all populations become polymorphic across the landscape, even though 2 out of 3 sites have a mutualistic interaction. Now, when the central community has a mutualistic interaction and the 2 peripheral communities present an antagonistic pattern of interaction, what happens is that within the central mutualistic community, interaction will remain high regardless of the degree of gene flow. And then for the antagonistic communities, what happens is there's a decrease in interaction, there's a decrease in polymorphism with a decrease in interaction. That decrease in interaction that we see right here in both communities means that there's a higher mismatch between those species. And finally, for moderate levels of gene flow, alleles become fixed for all populations. As we increase the meta-community size, what happens is that effect of the central community becomes more and more preeminent. So for a community where for a meta-community with 4 neighbors, and the central community being antagonistic, for even low levels of gene flow, all landscape will become polymorphic. In the same way, for a meta-community with the central community being mutualistic and the peripheral communities being antagonistic, all landscape becomes monomorphic with fixation of alleles for all species. So I have shown how gene flow and meta-community structure can influence coevolution between a pair of interacting species with variation in the outcome of the interaction. First, we can see that all else being equal, with a simple metapopulation framework with only two communities linked by gene flow, what happens is that both populations become polymorphic. If you remember, the mutualism would lead to fixation of alleles, and just a small value of gene flow is enough to destabilize those mutualisms, which suggests that mutualisms are more fragile than antagonistic interactions. Also, a central patch in a meta community will determine the result of the phenotypical patterns found in this species. And if we think about it in a conservatory, in conserving species and ecosystem services, what happens is that as we increase connectivity between patches, we can swamp what happens in local isolated patches. And finally, variation in community context and among communities, and how this variation is distributed within the landscape, creating those mosaics, will affect coevolution and will be important in determining how coevolution patterns, how evolutionary processes will generate the patterns that we see. With that, I would like to thank those guys that helped me develop all this, the funding agencies, and